Hello viewers, welcome to my Mad Scientist laboratory. Today I'll be conducting one of mankind's most important scientific experiments, the cloning of humans. I will take a completely willing test subject and create a duplicate of him. Now, how do I plan to do this? Why, with the same strategy we used way back in '96 to create Dolly the sheep. And it's called Somatic Cell Nuclear Transfer, or SCNT for short. And it goes a little something like this. Here I have a diagram of the basic principles of SCNT. There are two sides to the story. There is an oocyte donor and a nuclear donor. Both of their names have been redacted for confidentiality's sake. But from the oocyte side, we start with some eggs removed from the donor. We take out the mitotic spindle with a micropipette, and we're left with an enucleated egg, which is just an egg without a nucleus, as you can see here. From the donor side, we remove some muscle cells. They're placed in a culture and grown into the G1 stage. They're going to be put into a pipette and transferred into the enucleated egg. You can kind of see it's right there. And then the egg and cell are fused with an electronic current applied to the egg when the cell is put into it. The embryo is cultured for 7 days, at which point the blastocyst forms. The embryo is transferred into the surrogate mother, and eventually there's the birth of the clone who is genetically identical to the nuclear donor. So as I mentioned before, it is this procedure that ended up with us cloning Dolly the sheep in 1996, but a little less well known was Sir John Gurdon, who in 1958 did practically the same thing with frogs. It was his insight that led us to cloning as we know it today. So to begin, I'm going to take the nucleus out of our egg cell, and now onto the extraction of the muscle cells from our test subject. So now I'll take a somatic cell sample from my test subject. Now I will take the muscle cell and inject it into the enucleated egg cell. This allows the egg cell to be reprogrammed with the somatic cell's nucleus. Now all that's left is a quick 9-month incubation period. So now in my hands, I have the clone of my test subject. You can't quite see the resemblance just yet, so let me put it into my age-up machine. Ta-da! A clone! And now, the clone shall meet the original. Alright, so it's not perfect. There must have been differences in the DNA methylation pattern of the clone while developed. With imprinted genes, even natural identical twins can be just slightly different. My theory is that the imprinting just went awry in this experiment, and that's why they look alike, but they're not quite a carbon copy. But that's good enough for our purposes. I hope you've enjoyed this video and this huge leap for humanity and the sciences.